Today I'll be discussing CAR T cell therapy. It's an immunotherapy mechanism that can aid in the combat against cancer cells. First, we'll start with the discussion about T cells. They are a type of lymphocyte that develop in the thymus gland, and they are an essential figure in the immune response. They have these T cell receptors on the cell surface that allow them to adhere to foreign entities. Once they bind to the antigens, uh, the antigens present on the cell wall, they can kill the antigen itself. The issue that arises is that these T receptors don't recognize or adhere to certain cells like cancer cells, and this may be due to the cancer cells develop modes of evasion. So this is where CAR T cell therapy comes in. It can target these cancer cells, but I'll start with discussing how the procedure is done. First, the first step is to extract the T cells from the patient. T cells are collected via a process called apheresis. It's a procedure where the blood is withdrawn from the body and one or more blood components, in this example would be the white blood cells, they're removed. The remaining blood is then returned to the body, and the extracted T cells are delivered to the lab for the next step. And so the next step we will talk about is to reprogram these T cells, and to do that, you will use a virus that contains DNA, and this DNA will produce these special receptors. They're called a chimeric antigen receptor, or CARs. Um, and this manufacturing process can take from 3 to 4 weeks on average. While this time, the patient will be receiving a bridging therapy that will keep the disease like lymphoma or leukemia at bay. The next step that we'll discuss is the multiplication of the T cell. Um, this T cell can be polyclonal stimulated with an APC system that are made up of these CD8 T beads and modified LV APC that will induce proliferation. When there are enough of them, the CAR T cells are frozen and sent to the hospital or the center where the patient may be treated. And it's important to note that a closed culture system will reduce the risk of contamination and will promote optimal expansion. But before the patient is given this treatment, they will be prepared, and to do that, they'll go through lymphodepleting chemotherapy. And this is a brief course that will reduce the level of white blood cells so their body can accept the infusion of CAR T cells. Turning the CAR T cells to the patient, they are thawed out because they were previously frozen. Um, Once they return to the patient's bloodstream, they naturally multiply in number. So these are the attacker cells that will recognize and attack cells that have the targeted antigen. And these cells may eradicate all the cancer cells in the body, and the CAR T cells themselves will remain in the body months after the infusions have been completed. This has resulted in like a long-term remissions for some type of blood cancers. One example would be the Chimeria. It was the first successful story of this new treatment. Um, and Here is an overview of what I just kind of went through. If you can pause it and read each individual one if you like, it may help you get a better image of how the process is happening. But I wanted to mention that potential candidates include children and young adults with a B lineage and adults with an aggressive form of B lineage lymphoma. And they're only eligible if they have relapsed from previous therapy, like just your usual chemotherapy. Um, and this is a great thing, a great therapy. Here's another image where it shows the normal T cell receptors, and then it shows the modified CAR T cell receptors, and they actually bind to certain types of cancer. There are some risks. They do exist. Two main ones are cytokine release syndrome, or CRS, and just neurotoxicity. CRS is due to the activation of the T cells after they're given back to the patient. They, when they're given back to the patient in the patient, they release cytokines, which give the patient symptoms of fever, low blood pressure, difficulty breathing, and difficulty handling fluids. The second risk would be neurotoxicity. May This is also caused by cytokines, but it affects the function of the brain. Symptoms may include confusion, difficulty with words, and seizures. Luckily, though, those two risks are 100% reversible. CRS symptoms usually appear after day 1 up until day 7, um, and neurotoxicity symptoms occur a little bit after that, but they can last longer up until day 14. Another thing that it may cause would be something called B cell aplasia. CAR T cells not only attack tumor B cells, but they also attack the individual's B cells. And so the patient may require immunoglobulin infusion for the rest of their lives. But other than that, uh, this is something great. It doesn't work with everyone. Not everyone responds the same. For example, something called CD19 escape can occur, and this just refers to leukemia cells that don't express the targeted proteins anymore, and so they've grown and adapted to evade CAR T cells, which is something kind of scary, knowing that they can keep up with our technology and therapy. But overall, it is a promising therapy with a lot of potential. Uh, the therapy is currently being worked on to be optimized so it can work with more individuals rather than just specific type of uh, blood cell cancers. And this is just a image of, a comparison of a normal T cell with a CAR T cell. Now, CAR T cells are chimeric, so it doesn't do it justice that these receptors are all similar in color. Some of them may be different, which allows it to adhere to a variety of things. It depends on the DNA that was modified with. And here are just some of my reference. And Dr. Blair, I'm just saying that it is okay for you to share this for your future classes if necessary. Thank you.